Hi friends, so welcome to our channel Bio 360. In today we will be discussing about some interesting important concepts in 9th standard biology CBSE, which comes under 9th standard science. We don't have a separate subject called biology. So before we move on to the concept, so I would request you to kindly watch the entire length of the video. I believe it will be very useful, very informative. Do share your views in the comment section, share it with your friends, and importantly, if you have any such concepts to be discussed, so please do let us know in the comment section or mail us. So we'll be happy to bring those discussion in this forum, right? So this is going to be the topic. That is chapter 5. Chapter 5, the fundamental unit of life. So what does this topic deal about? This topic deals about the cell, which is the fundamental unit of life. So we all know that the cell is going to be the fundamental unit of life. So if you look into the entire topic, the various structural aspects of cell with its function would be given. Like what is a cell, what is its general structure, what is a prokaryotic cell, what is an eukaryotic cell, etc., etc., and what are the organelles present, what is the function of organelle. So the basic fundamentals are given. And let me be frank, most of the lines given in this are friendly, which means that once you read, you will be able to understand. But we do have certain areas here which are just given without any background information for us to understand. Okay, so our idea is to support those lines with the required background information so that you would find it really easy or helpful once you read these lines. So those informations will be really helpful, okay, with which you can understand these lines comfortably. So that's our idea. Okay, so when you go through this chapter, you would see that the introduction about the cell are being given. So here you see a structure of a microscope, because cells are microscopic. So microscope is required for us to observe the cell, and the historical perspective of the cell, like who discovered the cell for the first time and what led to his discovery. So all those things are given, and the structural aspects of cells are given. So our area of discussion here is this here is the one. Okay, yeah, plasma membrane or cell membrane. So this is our area of discussion. Sorry. Okay, so this is our area. So what is that to be discussed in plasma membrane? Yeah, so before which we all know that the plasma membrane is going to be the outer membrane. That is, it is universally present in all organisms except viruses, right from a bacteria till a animal, right? All organisms do have cell membrane or plasma membrane. It has got a very important role. But in certain organisms like plant, bacteria, fungi, it is the cell is, that is the plasma membrane is supported with additional membrane, which is cell wall. So you know all this information. My point here is the plasma membrane or cell membrane is very unique and very important. It is present in all organisms, right? This is the point. So what role does it play? It is like, I always say it is like a compound structure for a building. Okay, that is, it is a compound for a cell. This outermost membrane, outer membrane of the cell. Thereby, it is going to exhibit its own permeability, okay, in deciding a molecule, whether a molecule can enter and exit a cell. So permeability comes, okay, as a part of discussion here. It's very important aspect of plasma membrane. One thing, okay, so here you would see those lines saying that the substance like CO2 excreted out by the cell accumulates in high concentration inside. So you do have these lines, okay, which is mainly focused on or dependent on the permeability nature of plasma membrane. So here we are supposed to know what is meant by permeability, what are selective permeable, what are semi-permeable, fully permeable, impermeable. Though these terms are much more simpler to understand, but there is a sort of discussion would really be helpful, in providing much more clarity to these lines. So okay, so this is one point. These lines are given, but an additional information is what is needed. Okay, one thing. And based on the permeability, the concept will, the permeability concept would lead to a process called as diffusion and osmosis, which is mentioned here. So you could see the page number 51. You see page number 51. Okay, so you see a mention about selectively permeable membrane called as osmosis, and further if you move down, you do see the movement of water across the plasma membrane is also affected by amount of substance dissolved in water, etc., etc. So high solute concentration. So you do have a lot of concepts here. The lines are given, but for us to understand and enjoy the meaning, okay, present in these lines, we are going to support you with some background concepts or fundamentals. So with knowing those fundamental, understanding those fundamental, your understanding, okay, these lines will be much more clarity, right? So this is the idea. So with this, we'll get back to our discussion. Yeah. So here, first we will talk about a concept called as solution. We will begin with this and we will slowly build up the concept. Then we will get back to the textbook and see whether we are able to understand those lines, right? So let's all assume that, so everyone would have prepared some solution. So let us all assume that we are going to prepare a sugar solution. So what do? You take a beaker. Let me label this as A. You take another beaker. I'm going to label this as B. So in household, you can assume this beaker to be a container or a vessel. What we have it in kitchen. Right. So what is my aim here? To prepare a sugar solution. Right. So what do? Let me take 1 liter of water. Okay. Obviously, if it is a solution, I have to get it dissolved in water, right? And I'm going to add some amount of sugar. So either you get it from your shop or might be readily available. Mostly it will be readily available in your kitchen. So take some amount. So let's say, let me assume it to be 1 gram. Okay, 1 gram of sugar. The sugar is basically sucrose. What the table sugar, what we use in our home household purpose is biochemically sucrose, right? We are going to add 1 gram. So I get a sugar solution, right? So basically the point is this solution is going to have 2 components. One is the solute component, other one is the solvent component, right? Now, how about B? I have taken one more vessel, B. How about B? I'm going to take the same volume of solvent, that is 1 liter of water, H2O, which is the chemical formula for water, into which say I am adding 3 gram of sugar. Okay, I'm going to add 3 gram of sugar. So both are sugar solution. So both is going to have a solute component. Okay, so this is going to be the solute. We call this as solute, and this will be the solvent component. So both have got a solute component and as well as a solvent component, right? Now let us understand certain things here. If you look at the amount, okay, or the concentration of solute, so what is this? This is going to be 1 gram in 1 liter. This is going to be 3 gram in 1 liter. Hope you all agree with me. So this is going to be the concentration of the sugar. 1 gram in 1 liter, 3 gram in 1 liter. Now the volume of solvent, okay, volume of solvent will be 1 liter here, 1 liter here. So what is your idea regarding the concentration of solvent, that is concentration of water? What do I mean by concentration of water? I mean concentration of water, that the free water availability. See, assume the same water, I'm going to add say 1 gram, my sugar gets dissolved, pakka. 3 gram, it gets dissolved. But what happens if I keep adding the sugar? What do you think? At one point, my solvent is not going to dissolve my solute. Except you all can even try with some basic amount of sugar and water. You can try. So I cannot dump some high amount of sugar on 1 liter of water. It's going to say fine. There's a point above which, okay, the solvent is not going to dissolve the solute. So what happens here when a solute is added? The solvent molecule that is present in the the solution is going to engage the solute molecule so that the solute molecule will get dissolved. Okay, but when all the solvent molecule are engaged, the further solute molecule which is added, okay, will not get dissolved. Okay, I hope you get this point. So which means that let us assume, say 5 gram of solute, that is sugar, will be the maximum dissolution point. Okay, see, that is anything you add above 5 gram cannot be dissolved by the solvent. Okay, whatever solvent molecule available in 1 liter can maximally dissolve 5 gram of sugar. So it's a simple assumption. It may not be a fact, but it's a simple assumption. So what happens here is, when you add 1 gram of sugar, my solvent molecule still can accommodate 4 gram of sugar to reach the maximal point. So here I have already added 3 gram of sugar, still 2 gram of sugar alone is left. So where is the free solvent molecule available more? The free solvent molecule is much more available in A, because I have added only 1 gram of sugar. Still my solvent molecule are free to engage another 4 gram of sugar molecule. Whereas in case of B, I have already added 3 gram. So my free solvent molecule availability is going to be very, very less. So which means that it can accept another just 2 gram of sugar molecule. So I hope this is what I mean as the solvent concentration. I hope you understood. So my point is, the volume of solvent is going to be same, 1 liter, but the solvent concentration is going to be more in A, because the solute here is going to be less, right? So it is going to be more here, it is going to be less here. Okay, so these three are the important point, that is the solute concentration. Okay, this is solute concentration, or the solution concentration is 1 gram per 1 liter, 3 gram per 1 liter. So this is highly concentrated, this is less concentrated, right? By which I mean it has got a less solute, it has got a more solute. Volume of solvent, water, is going to be same in both the cases, but the concentration of solvent is inversely proportional to the amount of solute. Less the solute, more the solvent concentration. Okay, more the solute, less the solvent concentration, but solvent volume remains same, right? I hope you understood. Okay, so keep watching these videos. Let's continue this discussion in another video. Okay, thanks for watching. Thank you.